Now, as we're watching the bacterial population increase in size, we notice that there's a certain pattern across all bacteria. No matter what the species is, as the population is growing, we see certain things occurring within the population. So not only is the numbers increasing, but there are certain tendencies within the population. We call this a growth curve. An increasing number, a hitting of a stability, and then a decline as the nutrients and things run out. How do we know this? Well, like I said in lecture 6.3b, we inoculate with a small number of bacteria into a large volume of broth, and then we let it go. We let it grow, let it increase in numbers, and then we sample. Every so often, at a specific time intervals, we sample and we count. Plate it out, do serial dilutions, plate it out, count the number of colonies, and then we calculate based on dilutions, based on the number of colonies. This is how many bacteria have to be there in the flask. Now, the colony counts, we call that CFU, colony forming units. Because as we like to think that each colony started off as one bacteria. 1 became 2, 4, you wait 24 hours. Exponential growth, 2 to the 30 is 1,000,000. You can't see one bacteria, one bacterial cell, but what you can see is one bacterial cell and 1,000,000 of its clones piled on top of each other. We call that a colony. We talked about that back in chapter 1, I believe. So when you go and you look at and you study how we do this, we plate out, we take a sample every 30 minutes, every hour. Plate them out, count the number of colony forming units, multiply it out by the dilution. See how many was there originally. What we notice is also the bacteria and the culture go through 3 or 4 phases. 4 phases that all bacterial cultures go through. We see that initially, for a bit of time, once you initially inoculate the bacteria into the new broth culture, there's very little growth. The number of bacterial cells dividing is about equal to the number that are dying. We call this the lag phase. The bacterial cells haven't quite responded, haven't quite started expressing new genes due to the availability of new nutrients, the availability of, whatever. So it's in balance. Given a little bit of time, half hour, couple of hours, depending upon the bacteria, we start to see the number of dying goes down and the number of dividing cells, thus the living healthy cells, starts to go up and up and up. We call this the log phase. The number of living, healthy, actively dividing, metabolically active greatly outnumbers and increasingly outnumbers the dying. Log phase, because it's exponential growth phase. The numbers 2 to 4 to 8 to 16, 32, 64, and on and on and on. Dividing cells greatly outnumber the dying cells. Lastly, or next, you get the stationary phase. The numbers have increased and increased, and well, you know what? Nutrients are starting to get a little depleted. Waste are starting to build up, so active division starts to taper a bit. The number of cells dying starts to increase. We're back to a balance. For every cell that's dividing, there's a cell dying. Now, keep in mind, the population numbers are, in the trillion, 10 to the 10th, 10 to the 11th, where you started out with maybe a few hundred, you're now a billion-fold higher, a trillion-fold higher. Then the last phase, the fourth phase, the death, decline phase. Nutrients are all but depleted. Problem here also is the dead and the dying cells and the waste products are polluting, for lack of better terms. Start to see the actively dividing start to go down. The dying starts to go up. So the numbers of living cells declines. As we see here, when we look at this graphed out, the lag phase, everything's just starting. The exponential growth phase, the log phase, the numbers are increasing logarithmically, 2 to the n. It just keeps going and going and going till it's the stationary phase where the broth just can't sustain anymore. This cannot sustain the exponential growth. It's basically reaching its limit. And it'll stay this way. For every one dividing, becoming new, there's another one dying. And then everything gets depleted. Waste built up too much, nutrients depleted, and the death phase, the declining phase.